Welcome to this presentation on Halo PT, a new proficiency testing program. Although I shall be focusing on fresh and frozen umbilical cord blood, this proficiency testing program will also be available for bone marrow and mobilized peripheral blood. Hello, I'm Ivan Rich, CEO of HemoGenix, and I shall be accompanying you through this presentation. So let's start by considering what proficiency testing is and how it relates to hematopoietic stem cell therapy products and umbilical cord blood in particular. NetCord Fact defines a proficiency test as one that evaluates the adequacy of the testing method and equipment and the competency of the personnel performing the test. Proficiency testing is a tool the laboratory can use to verify the accuracy and reliability of the testing. Accuracy and reliability are parameters measured during the validation procedure, but proficiency testing is also a method of interlaboratory comparison and therefore measures assay robustness, which is part of the assay validation procedure. I shall be focusing on cord blood proficiency testing. However, the same aspects I'll be discussing apply also to mobilized peripheral blood and bone marrow. Since bone marrow transplantation became a routine procedure in the 1970s, product testing has been based on minimal criteria, recommendations, and standards. This is especially the case for cord blood. One has to be fair and say that there were no other alternatives available in the 1980s and 90s. However, that has now changed dramatically and is even more acute since cord blood started to be regulated as a result of the Stem Cell Therapeutic and Research Act of 2005. The minimal testing criteria include a total nucleated cell or TNC count, viable nucleated cells, viable CD34 positive cells, and a CFU or other validated potency assay. Notice that with the exception of the CFU assay, all are performed post-processing and pre-cryopreservation, rather than post-thaw and prior to use. The CFU can be performed at any time apparently, prior to release, but is usually performed prior to cryopreservation. It just so happens that potency assays must be performed prior to use of the product in a patient. So here we have a considerable amount of ambiguity, even from the FDA. Notice also that the NMDP CFU requirements for the CFU which must be reported as growth or no growth with an actual colony count. But this count can be either the total number of colony types or just a granulocyte macrophage colony count, which is also ambiguous and really provides no proper information. Now, there are proficiency tests for all of these minimal testing criteria. However, one of the reasons for introducing a proficiency test for the CFU assay was due to the inherently high coefficients of variation, or CVs, seen with the assay. These are due to a number of factors. The first is the use of a TNC fraction rather than a mononuclear cell fraction, or MNC fraction. The use of non-standardized methods for adjusting the cell concentration, the inherent difficulty of accurately dispensing methylcellulose, the lack of standards and controls, the lack of interlaboratory colony counting consistency, and the lack of assay validation. All of these factors contribute and add to the variation seen in the CFU proficiency test. This is the type of proficiency test used by many cord blood banks and transplantation centers using the CFU assay. They're sent a cell sample, which is then set up in the assay, and the number of colonies is counted after 14 days. The first thing to notice is the range of colony counts. This goes from 0 to 150, with a mean number of colony counts at around 45. If we look at the number of colonies that make up a one standard deviation, this is about 22, which means that one standard deviation is equivalent to CVs of greater than 48%. This demonstrates why few relevant statistics can be obtained from the CFU assay. The information you as a user obtain from such a proficiency assay or test is essentially zero, and therefore a waste of time and money. Similar results were published by the NMDP in 2011 that included not only the CFU assay, but also CD34 enumeration. The very high CVs only tell you that the tests and assays are essentially unacceptable for clinical use. So after 25 years of minimal cord blood testing that does not even determine the characteristics of the cord blood stem cells responsible for engraftment, and required by the Stem Cell Therapeutic and Research Act of 2005, and have not improved the 15 to 24% graft failure seen with cord blood transplantation, HemoGenix believes it is time to initiate best practices for cord blood testing and other hematopoietic stem cell products. HemoGenix has developed a family of assays specifically designed for hematopoietic stem cell therapy processing applications, especially cord blood testing, that encourage best practice criteria rather than just minimal testing criteria. These assays include Halo 96 PCAEQ, which is a CFU equivalent assay using similar growth factor cocktails to Methocult, for fresh or frozen cord blood performed in a 5-day assay. StemPredict is a predictive functional 3-day stem cell assay to determine bankability on fresh cord blood prior to cryopreservation. Halo 96 SBCQC is a 5 or 7-day assay to determine stem cell proliferation ability, also called stem cell quality, before and after a procedure. Halo 96 PQR is a 7-day FDA-compliant and reference standard-based cord blood stem cell potency, quality, and release criteria assay performed on a segment prior to transplantation. And finally, Halo 96 PMT is also a 5 or 7-day assay to predict time to engraftment and to monitor global patient reconstitution. These are described in detail on the HemoGenix website and as videos on the HemoGenix YouTube channel. And to ensure that these assays are performed correctly and accurately, we've introduced a proficiency testing program. This is called Halo PT, or Halo Proficiency Testing, which promotes best practices for cord blood testing, as well as for other hematopoietic cell therapy products, by providing the most advanced proficiency test available. Halo PT is a calibrated and standardized 7-day proficiency test assay that measures the proliferation of hematopoietic stem cells in a dose-dependent manner using the most sensitive and accurate assay readout available, namely ATP bioluminescence. The ATP bioluminescence technology is described in another HemoGenix YouTube video. Halo PT is performed in 3 steps. First, the assay is set up and the cells cultured from fresh or frozen mononuclear cells. Second, there's a calibration and standardization of the assay. And finally, there's the measurement of cell proliferation of the sample. The results are exported from the instrument, a luminescence plate reader, to an Excel file and sent to HemoGenix for analysis. The results from each participant, as well as an interlaboratory comparison, are returned. A certificate of proficiency is provided, as well as recommendations on how to improve accuracy and proficiency. The benefits of performing Halo PT are shown here. Halo PT ensures that stem cell therapy samples are measured to the highest accuracy and competence to perform any Halo assay. It ensures that the instrument is set up correctly and functions properly. It demonstrates proficiency in dilution and dispensing techniques. The assay ensures that the basic stem cell quality and potency can be measured. It provides a two-layered proficiency test with predefined acceptance-rejection criteria by quantitatively comparing instrument calibration and standardization techniques and cord blood stem cell characterization used for potency and quality determinations. HaloPT takes about an hour, 30 minutes to set up the assay, 30 minutes to calibrate, standardize, and measure the sample, and then 7 days to culture the cells. A luminescence plate reader is required to perform HaloPT, or any of the other ATP bioluminescence assays. The instrument costs less than $10,000, and it has to be connected to a PC or Mac running Windows. A good microscope will cost a lot more. If you're considering automating colony counting, you'll be spending 5 to 10 times as much money, and it will only be used for a single assay. So a luminescence plate reader is an extremely cost-effective investment that can be used for multiple applications and helps in promoting best practice criteria. Now, unlike the CFU assay or other minimal tests, HaloPT quantitatively measures a number of important parameters. These are divided into two levels. The first level is the ability to calibrate and standardize the assay. So here we're going to be measuring accuracy of the expected ATP standards to dilutions, coefficients of variation for ATP standard expected results, the slope and correlation coefficient of the ATP standard curve, accuracy of the controls, and control value CVs. The second level involves the ability to measure cord blood stem cell proliferation. And this will entail the ability of the cells to be stimulated and proliferate, ATP values for each cord blood stem cell dilution, sample cord blood stem cell CVs for each cell dose, and the slope and correlation of the linear regression of the cord blood stem cell dose response curve. You'll see the importance of these in just a moment. So let's first take a look at how HaloPT is set up. Cells for performing HaloPT will be shipped fresh 1 day prior to the study so that they arrive on the day of the study, or as a frozen sample which should be stored in liquid nitrogen until the day of the study. All cells used in the assay are mononuclear cells, or MNCs. A TNC fraction cannot be used because it contains red blood cells, granulocytes, and platelets that dilute and mask the stem cells. Please remember that this is a stem cell assay for the cells responsible for engraftment, not a progenitor cell assay. On the day of the proficiency test, the frozen cells are removed and they're thawed in a 37-degree water bath. The actual procedure for handling fresh and frozen cells is described in detail in the HaloPT assay manual, which is included with the kit. After thawing, DNase is added to reduce clumping and stringing that might occur as a result of cells dying. The cells are mixed gently with a pipette. Fresh MNCs can be used directly after measuring the nucleated cell count. The frozen cells are gradually added to an IMDM or IMDM medium in a tube and mixed gently by inversion. The thawed cells are centrifuged at 200 times G for 10 minutes at room temperature. The supernatant is discarded and the cells resuspended in medium. Fresh cells are not washed prior to use. A nucleated cell count and viability is determined. The cell concentration is adjusted to 400,000 cells per milliliter, and a serial dilution to 200,000 and 100,000 cells per milliliter is performed. This is shown as the 3 tubes on the right-hand side of the rack. On the left-hand side of the rack, 0.9 milliliters of the Halo PT master mix has been dispensed into each of another 3 tubes. 0.1 mils of the lowest cell concentration is then dispensed into the Halo PT master mix tube for that cell dose. This is repeated for each of the other cell doses. The procedure dilutes each cell concentration tenfold. Once each of the cell doses has been added to each of the Halo PT master mix tubes, the contents are mixed and 0.1 milliliters is dispensed into each of 8 replicate wells of the sterile 96 well plate provided with the kit. Starting with the lowest cell dose on the left-hand side, all 3 Halo PT culture master mixes have been dispensed. Each of the cell concentrations has been further diluted another tenfold to produce final cell concentrations of 1,000, 2,000, and 4,000 cells per well. It is extremely important that 8 replicates for each cell dose is performed. The lid is replaced and the plate transferred to the incubator for a 7-day culture period. So after 7 days of culture, the assay is calibrated and standardized as described in the HemoGenix YouTube video setting up the ATP standard curve and controls for all HemoGenix ATP bioluminescence assays. Prior to measuring the samples, the first level of proficiency testing is performed. This involves calibrating the instrument and performing the ATP standard curve to standardize the assay. Calibrating the instrument is performed using the controls which must lie on the ATP standard linear regression curve and be within specific predefined acceptable ranges. Standardization of the assay is performed using the ATP standard curve, which must be within a specific slope value range provided by the B parameter shown underneath the graph. The goodness of fit, or R squared, must be greater than 0.995. In this case, it is 1. In addition, the coefficients of variation, CVs, must also be 15% or less. This is required by FDA guidelines. The final practical step for performing Halo PT is shown in the HemoGenix YouTube channel video, How to Process Samples and Measure ATP Bioluminescence for any HemoGenix assay. The second and final level of proficiency testing is shown here. The results that you might get are shown for 2 different participants. Here you see the 3-point cell dose response curves for the same cell population determined on the same lot of thawed human umbilical cord blood stem cells. The parameters that are measured and compared are first the ability of cells to be stimulated and proliferate. That must be greater than a predefined acceptance rejection value at 7 days of incubation. The slope of the dose response. This measures stem cell proliferation potential and is used to evaluate the potency ratio, which is required for product release. The third measurement is the ATP concentration values at each cell dose. This is used to measure stem cell quality and also required for product release. Finally, the coefficients of variation, or CVs, at each cell dose are measured, and these are used to determine the accuracy of cell dilution and dispensing, and must be within a predefined range. So in summary, after performing Halo PT, the participant will be able to perform any Halo assay accurately and efficiently. The participant, laboratory director, medical director or transplant physician, and patient will know that results from any HALO cell therapy assay used to determine the stem cell characteristics of the product have been performed to the highest level of competency and accuracy. After 25 years of minimal testing for cord blood transplantation and longer for other hematopoietic stem cell therapies, these criteria can now be supplemented with proficient, quantitative, and validated assay results as required by the Stem Cell Therapeutic and Research Act of 2005. HALO PT is the key to promoting best practices in hematopoietic stem cell therapy by demonstrating proficiency in performing stem cell quality and potency assays necessary to providing the patient with high quality and a potent stem cell product. So thank you for your participation, and please visit our website for more information on best practices and the HALO PT program. Bye-bye.